So let's jump into chapter 2. In chapter 2, we are going to talk about the structure of genes and DNA. And then we're going to talk about how genetic information goes from, how our genetic information is used from the code all the way out to producing a particular phenotype of an individual. We are going to focus most heavily on 2.2 and 2.3. Only going to talk very briefly about structure and function of genes and gene regulation, and then we are going to skip transcription profiles. Some of this we'll come back to a little bit later. So the main focus here in this chapter is going to be the structure of DNA and then the central dogma, which is the process of gene expression. So in the between the early 1900s and the 1950s, chemists and biologists started looking at the structure of DNA. And there was a lot of question about whether the genetic material that was passed on was DNA or protein. And so we'll talk about that a little bit later. But before you can really figure out how it works, you have to figure out what it is. And so in the 1950s, there was a lot of research and thought going into what the actual chemical structure of DNA was. Now, two important researchers at this time were named Wilkins and Franklin. Maurice Wilkins there on the left, Rosalind Franklin on the right. And they were using a technique called X-ray crystallography. Now, X-ray crystallography is a process by which you shoot an X-ray at a crystallized molecule of some sort, whatever you are interested in studying, and then you analyze the diffraction pattern of those X-ray beams. So X-ray beams hit the crystal, they are diffracted into a particular pattern onto a sheet of film, and then each crystal will always produce a particular diffraction pattern, and from that pattern you can deduce the shape of the molecule. And so Rosalind Franklin was doing a lot of work on this. This picture in the middle is a very famous picture called Photo 51. And this is a photo that is actually taken from sort of the end of the DNA looking down, almost like you were looking down a tube or a straw, something like that. And so the way that this works is you look at the distances in the diffraction pattern, and you measure these distances, and from those distances you can determine the overall shape of the molecule based on bonding angles and things like that. But they were struggling with this. They couldn't quite figure out what was happening. They thought that it was probably a helix of some sort or a twist, and they thought that it probably had two backbones, but they couldn't quite figure out what exactly was going on. So two other scientists, James Watson and Francis Crick, who you've probably heard about, became interested in this as well. And they took Franklin's images. It is said that they took them without her permission. And that's a whole nother story we could go into. But they took some of her images along with their own knowledge of X-ray diffraction and started building models. So they took the X-ray diffraction images and calculations from those images and started to apply them to a physical structure. And we've got this picture up in the top right that shows their model of the structure. They were literally using chemistry models. They were not using anything fancy, and they were basically trial and error with some input, things that they already knew. And they deduced that the overall structure was a double helix with two backbones on the outside held together by molecular structures in the middle. That double helix structure is made up of repeating units called nucleotides. Now, these nucleotides are what give DNA its name, its chemical name. DNA stands for deoxyribose nucleic acid. So that's a mouthful, but let's talk about what that means. So deoxyribose nucleic acid is made up of subunits called nucleotides, or deoxyribonucleotides, DNTP for short. That S there just means multiples. It doesn't need to be capitalized. So each of these nucleotides is made up of some specific parts. The first part, or the sort of the base of the structure, is the deoxyribose. Now this is a sugar, specifically the sugar ribose, and it is a 5-carbon ring structure with an oxygen in the middle. So this makes a pentagon shape. You'll notice that this sugar is labeled with some numbers. So when we number our carbons, these are numbered carbons. So each of these is a carbon here. And we number our carbon around the ring. So the first one after the oxygen is the 1' carbon. You'll notice that we have an appendage sticking off of that 1' carbon. We have the 2' carbon. Now this carbon's important because ribose is actually supposed to have a hydroxyl group, or an OH at this spot. But you'll notice that we are using deoxy, or ribose without an oxygen, and that oxygen is missing from that 2' carbon. You'll also notice that there is a oxygen and a hydrogen off of this 3' carbon. This guy's going to be important in just a minute. We're going to skip over that 4', and there's a 5' carbon, which is actually the carbon sticking off of the ring, and attached to that is another part of our molecule. So the basis for our DNA molecule is this deoxyribose sugar. Attached at that 1' carbon is the nucleotide base, or a nitrogenous base. This is a chemical structure we'll see in just a minute that is made up of nitrogen and carbon rings. And it is these bases that give us our genetic code. That code is written in a 4-letter code using 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. So those 4 bases are going to be attached to that ribose sugar. So we've got the D, the deoxyribose, the N, the nitrogenous base. Let's talk about that third part, all those phosphates sticking off the side over there. So the TP part is triphosphate. This is this arm sticking off of that 5' carbon, made up of 3 phosphates and a whole bunch of oxygen. Now you'll notice that those oxygens are negatively charged. This gives DNA an overall negative charge to the molecule. And this phosphate group is going to be important in linking our nucleotides. So let's start to build our DNA molecule. Now we say that DNA is built in a particular direction, or that it has a polarity. The direction of our DNA is going to be 5' to 3'. That means that we are going to attach molecules at a particular location. The only place that we can add a nucleotide is to that 3' end. So we are going to do this by linking the 5' molecule, or the 5' end of an incoming nucleotide molecule, to the 3' end of the existing molecule. When that happens, we're going to release a couple of phosphates, and we are going to form a bond between the 3' end and the 5' end of the next molecule. As we continue to do this, we are going to grow our nucleotide chain. So let's bring in another molecule. We have an empty 3' end. We are going to add that 5' end to the OH there. And when we do that, we are going to incorporate that phosphate group into the bond between our nucleotides. That's going to be important in a minute. So if you look at this molecule, we actually begin to see our 5' to 3' direction. At this end, at the top in our image, we have a free 5' end, and at the bottom, we have a free 3' end. That means we can continue to add our nucleotides, or DNTP molecules. So our growing chain continues to have that 5' to 3' polarity. So our DNA molecule also has, the overall molecule also has a 5' to 3' direction for each of its chains of molecules in the double helix. As we begin to grow this nucleotide chain, we start to produce what we call a sugar phosphate backbone. So as we link our nucleotides, we are forming covalent bonds between our 3' and 5' end. Now, covalent bonds are important because covalent bonds give stability. So the stable bonds on the outside on the backbone of our DNA molecule is going to make it a very stable molecule altogether. Now you'll see that the backbone is alternating sugar and phosphate molecules. Phosphate, sugar, phosphate, sugar, phosphate, sugar. And then the nitrogenous bases stick off into the middle. Now keep in mind that we're going to make a double helix out of this. So the other side is going to be a backbone with bases sticking off as well. Now remember that I said that those phosphate groups were negatively charged. That means that our sugar phosphate backbone is also going to have a negative charge. And this is what makes DNA an acid. Remember, deoxyribose nucleic acid. Now this is important because it affects the way that it's going to interact with other molecules. Most importantly, water, because we have a lot of water within our cells, so DNA is going to interact with water. Remember that water is negatively charged, so they behave nicely together. It's going to affect the way DNA interacts with proteins. Proteins can be positive, they can be negative, or they can have no charge. And depending on their charge or the region, the specific region of the protein can have different charge as well. We're going to have different interactions with proteins within the cell. And then, of course, this is also important for our laboratory procedures. So you may have done gel electrophoresis in your intro biology course. I hope that you did. That is a laboratory technique that is going to utilize this idea of the negatively charged backbone. Remember that the overall structure of DNA is a double helix. Watson and Crick so wonderfully deduced this for us. And the double helix actually utilizes the 5' to 3' directions as well. So remember that whenever we have a free 3' end, we can add a new nucleotide. So on our strand on the left, we actually have 5' at the bottom. So in this case, we're running 5' to 3' bottom to top. On the other side, we're running 5' to 3' top to bottom. Now in our cells, our DNA is not running up and down any particular direction, but each end is going to have a different polarity. We say that these strands are anti-parallel. They run parallel to one another in opposite directions. So our free 3' end is on different ends depending on which side of the backbone we're looking at. Now, if we make this into a cartoon model, we can see the 5' to 3' a little bit easier. When we take out all of those molecules, we can see things just a little bit easier. So we've got the 5' to 3' backbone and 5' to 3' backbone on the other side in the anti-parallel direction. Now, we haven't talked about those nitrogenous bases in the middle too much yet. But we did say that those are what gives us our code. So we have this thymine, guanine, adenine, thymine, all the way down. That is the beginnings of a genetic code. Now you'll see that those bases are connected in the center. That's another important feature of our DNA molecule. All right, so one of the important things about that DNA molecule are the bases, the nitrogenous bases that are connected in the middle. Now you might notice something peculiar going on here. You might notice that cytosine and guanine are always paired and adenine and thymine are always paired. This is called complementary base pairing. So guanine is always paired with cytosine and adenine is always bound with thymine. Now this produces some regularity in the DNA molecule. Now we see regularity in two places. One is in the turns of the helix. If you look at the distance from the start of one turn to the end of that turn, it's always the same distance. So each individual turn always measures the same length. The distance between sort of the pocket and the outside of the backbone is regular as well. So every time you have this little pocket, it's going to be the same distance. And then the distance between the base pairs is always the same. Now all of this is related to bond angles and how the molecule is actually put together. We're not going to go into all of the chemistry for that. The other important feature of the DNA double helix is that it has a uniform width, and that has to do with that complementary base pairing. We'll talk about all of the nitrogenous bases in more detail in just a minute, but when you combine two bases in the proper way, cytosine to guanine or adenine to thymine, you get a uniform width all the way across the molecule. If you were to bind together the wrong nitrogenous bases, you can get a bulge or a waistline essentially in your molecule. So if you don't pair your bases correctly, you're not going to have a uniform width in your molecule. So we get uniform structure in the distances between our base pairs, the width of the molecule, as well as the turns in the molecule. So let's talk a little bit more about these nitrogenous bases. Now these nitrogenous bases are complementary, which means the same bases always bind to one another. But these nitrogenous bases are broken into two types. The first type is the pyrimidine. The pyrimidines are the thymine and the cytosines. Now I remember this because they all have Y in the name: pyrimidine, thymine, cytosine. Thymine and cytosine are made up of single rings. So those rings are made up of nitrogens and carbons, but there's only one ring in their structure. And you can see they are similar, except that cytosine has this extra functional group sticking off of the side. Your purines, the other group, are made up of two rings or a double ring structure. Now similarly, they are made up of a 6-carbon ring and a 4-carbon ring, carbon and nitrogen I should say, but both of them are made up of those two rings, and then again they differ in their functional group attachment. The reason that these pair in a particular way has to do with the way that the bases bond with one another. So you will notice we always bind a pyrimidine with a purine. And we always bind a thymine with an adenine. Now, binding a purine and a pyrimidine is what's going to give us that uniform width, because if you look, we will have 3 rings all the way across. And if you go back to our complementary base pair page, you'll see what I'm talking about with that. But if you bind a purine with a pyrimidine, you get 3 rings all the way across. Thymine and adenine bond with 2 bonds in the center, and this type of the bond here is called a hydrogen bond. So adenine and thymine produce 2 hydrogen bonds, and cytosine and guanine produce 3 hydrogen bonds. So the importance of this is that when you have thymine and adenine, you have the correct number of bonds and you have a stable bond. When you have cytosine with guanine, you have 3 bonds, the correct number of bonds between those 2 molecules, and you have stability. If you bind something that's not supposed to be together, let's say thymine with guanine, you're going to have a bond that's not actually made and you're going to have instability. You're also going to have although you might have the correct width, you're going to have some trouble with that part of the molecule. And this is important because incorrect bonding can be corrected. And this is one way that the cell will correct mistakes, is by identifying incorrect bonds. Now, another important feature of these double and triple bonds is that if you have only 2 bonds, you have slightly weaker pairing and slightly stronger pairing between the molecules that form 3 bonds. So when you have cytosines bound to guanines, you have to have more energy in order to pull them apart. And this is important when we start talking about replication and gene expression, when we are actually going to pull our double helix apart. So in regions where you have a lot of adenine and thymine, you actually require less energy to break those bonds and pull the molecule apart at that location. So complementary base pairing is important for a lot of reasons, and it is actually probably the most important principle in molecular biology. Now, the reason is that if you know one side, you know the other side. So you know the code on the complementary strand. This means that you always have a template and you can always make the second strand. So that is important in DNA replication, where we're going to use one side to make a copy to produce the other. It's important in transcription, which is where we are going to take the DNA information that we want in a particular gene and use it to make a protein. It's important to translation, where we actually read the genetic code and make that protein. And it's important in many laboratory techniques, one of which is restriction enzymes, where we can actually cut DNA at specific locations. Restriction enzymes are actually a natural phenomenon found in bacteria, but we use them in the lab quite often. Complementary base pairing is also important when we're doing DNA sequencing, so we'll talk about that a little bit later, and hybridization, which we'll talk about in just a minute. It's important in PCR, which is basically in situ or in the laboratory DNA replication. Gene editing, we can use the current sequence to make or edit a new one. And then lots of other laboratory techniques also utilize this idea of complementary base pairing. I would say that the single most important idea, though, is DNA replication, where you always have a template with which to make a new copy of DNA. So let's talk about one of those techniques that utilizes this complementary base pairing. This is a laboratory technique called hybridization, and it is used in a lot of different ways in the laboratory, and you'll probably see this when we start talking about different laboratory techniques. But hybridization in general uses a short piece of DNA to tag or identify a larger one. So this short piece is called an oligonucleotide, and this is usually something that's made in a laboratory. So you might say, I want to find this particular sequence. I want to see if this sequence is present in this sample that I have. And so you would engineer and purchase this short segment of DNA. And then with the sequence that you want. And then you would have your target DNA, some sort of DNA that you have taken from an organism of interest, bacteria or humans or whatever you're interested in. And you are going to use this by first denaturing your strand. Denature means to pull apart, and you do this using heat. It breaks those hydrogen bonds between your strands. So you pull your strands apart, and then you allow your oligonucleotide to hybridize with your target DNA. Now, we can use this for PCR, or polymerase chain reaction, which I said was replication in a bottle, basically. So replication in a lab. You can also use this to identify sequences of interest. So if you think you have a gene in a particular species, but you don't know for sure, you can use an oligonucleotide with a short segment of that gene and see if it binds to the DNA that you're looking for. So you can actually use this technique to determine if a gene is present. You can also determine where it is present and if it is present in multiple times. So this is a really useful technique, and we will actually use this idea several times in lab this semester.